All organisms are composed of molecular machines, proteins, that perform most every biological function. Proteins are large molecules composed of chains of amino acids that fold into complex structures that determine their functions. Proteins are formed in the body when DNA is transcribed to form RNA, which is then translated by ribosomes and folded into their proper shapes. In the body, a delicate balance of protein synthesis, proper protein folding, and degradation are necessary to ensure that biological functions occur. If any of these steps are done incorrectly, biological functions can be disrupted, leading to diseases such as Alzheimer's or cancer. Understanding how proteins are expressed and function is hence essential to understanding how to prevent and cure disease. Researchers can study how proteins are expressed and function using proteomics. Proteomics studies the expression of proteins in the body by quantifying relative protein abundances in biological samples using mass spectrometry. Unlike genomics or transcriptomics, proteomics is an accurate measure of changes in expression. As a result, proteomics reflects how protein expression changes between disease and health states. Hence, proteomics helps bridge the gap between genes and expression. Traditional proteomics, however, struggles to achieve the full aim of bridging genes to function. Quantifying abundances of proteins allows researchers to understand changes in expression associated with disease, but can fail to capture the biophysical roots of these changes. For example, many of the least understood diseases, such as Alzheimer's or Parkinson's, are characterized by aggregation of misfolded, dysfunctional proteins. To a mass spectrometer, a misfolded or properly folded protein are very similar, if not the same, meaning that quantifying abundances does not identify differences in structure and does not provide enough information to accurately describe the mechanisms or roots of these diseases. As structure determines function, structural proteomics methods are necessary to bridge the gap between genes and function. Advances in the field of structural proteomics, such as the Iodine Protein Stability Assay, or IPSA, can help remedy this gap. IPSA works by partially unfolding proteins using a concentration gradient of denaturants. As proteins partially unfold over different concentrations of denaturant, amino acids become exposed to solvent. These exposed amino acids can then be labeled using labeling reagents such as iodine. After they are labeled, samples are then digested like in traditional proteomics. After digestion, labeled peptides can be quantified using mass spectrometry. Using curve-fitting software such as CHAF, protein folding stability values, CHAF values, can be quantified for each labeled site, painting a picture of stability across regions of proteins. This allows IPSA to identify differences in protein structure at a residue-specific resolution and a proteomic scale, allowing for high-throughput screening of structural differences. Unlike traditional structural biology approaches such as X-ray crystallography, IPSA can also be performed on complex biological samples under physiological conditions, allowing for a biologically accurate measure of structure. IPSA has also been demonstrated to be able to identify differences in stability of protein domains due to factors such as aging, mutations, ligand or drug binding, and presence of disease. These applications enable IPSA to be a powerful tool for research, diagnosis, and drug development. By applying IPSA and similar structural proteomics approaches, researchers will be able to bridge the gap between omics and function, empowering a greater understanding of human health, disease mechanisms, and drug and intervention development.